Hello, everybody. It is time to talk about one of my favorite topics in the brain-body connection, and that is signal transduction in the eye, which involves two distinct types of modified neurons referred to as rods and cones. One of the other reasons I like this, besides the interesting pathways that they use, is that they're named for their shape. Rods were named rods because they look like rods here at the top, whereas cones are named such because they look like a cone. What a great idea, naming things after their shape or appearance. When we think about photoreceptors for cells, again, we are dealing with these two types of modified neurons. They are neurons in that they are cells that respond by having somewhat action potential. We'll see that in a second. But they convey their information through nerves to the brain. These are called photoreceptors, photo referring to light. These receptors respond when they detect specific wavelengths of light. These are located in the neural tunic of the eye, and they are held against the back of the eyeball by the thick gelatinous vitreous humor. And when they are held in that area, they are in contact here in the fovea region of the retina. They're held in contact with their blood supply. So if they detach for any reason, so if the retina detaches from the eyeball, we now lose vascular supply to the rods and the cones, which means they can degenerate. They can also be destroyed by really intense light, like why you're not supposed to look directly into the sun or look at a solar eclipse while it's happening, because we can mechanically cause destruction to these rods and cones as well. These sensory portions, these outer segments, they are renewed about every 24 hours. So they're continually going through a mitotic division process. And when those tips are broken off, they are phagocytized by cells in the area so that they can be replaced by fresh, new outer segments of both the rods and cones. As I mentioned, these are modified, specialized neurons, and their receptive region, so the area by which they receive information, is at a portion called the outer segment. And the outer segment lies in front of or above the nucleus of these cells. So we see some typical cell portions. We see a dendrite region down here. We see a cell body in the center. But then at the outer segment, we see a specialized area meant to respond to wavelengths of light. In these outer segments, which are stacked like discs, that's what you see there on those lines, are photopigments. Photopsins are what you'll find in cones. We'll see that written on a couple of slides down. And rhodopsin, like rod, is the pigment that is inside of rods. So photopsin in cones and rhodopsin in rods, it is a physical photopigment. So just like you use a pigment as a dye, these photopigments exist in the outer segments, and they respond to specific wavelengths of light. Looking at a rod first, rods are, if you want to think of what they do, they are important for vision in the dark. And when you are in the dark, including when your eyelids are closed and you're sleeping or just, closing your eyes for a second, that's when rods fire. They are very sensitive to light in that when there is a light turned on, rods need to turn off because their job is to transmit information from a dark environment in the form of a simple black and white to gray, blurry picture to the brain so that you can make out more fuzzy types of pictures when you're in a dark environment. So they work mainly with night vision, and they're also active in peripheral vision, which is seeing things off to the side of you at the very limits of your field of vision. That is where you'll see the light being focused on an area a little bit outside of the fovea, where you have very high concentrations of rods and cones. So these rods, again, we see our outer segment, stacked discs filled with photopigment that will respond to light. Here's our inner segment, which connects the outer segment to the cell body here. We see mitochondria in the rods that are important for generating ATP for cellular responses, and we see a typical nucleus and the other cellular organelles not pictured inside of these rods. When we look at how rods are organized in a little bit, we'll see that many rods converge with the next level of cell to connect many rods to these optic nerves. What that is called is a converging pathway, many rods per one nerve. So humans in particular have a lot of convergence in rods, which is why we're not very good at seeing in the dark. However, animals that are nocturnal, they generally have a low converging pathway, meaning they have one rod to one neural cell. So it's a 1-to-1 ratio. And when there's not a lot of convergence like you see in humans, we get really clear, distinct pictures. So you would see that in nocturnal animals. Cats, for example, have a 1-to-1 rod to bipolar cell ratio. Humans, we don't. The brother or sister to the rod is the cone, and cones, the C in cone, should help you remember that these are responsible for color vision. Cones turn on when there is a bright light. They have a pretty low sensitivity. So if you think about being in a completely dark room and then all of the lights turning on at once, you're like, ugh, and you get that little glare where it's hard to even keep your eyes open. It's aversive. It hurts. That happens because all of the cones turn on at once, and that is called a glare when they're all stimulated at one time. So they react very quickly. You don't need the eyes to adjust when you're in a dark room and you flip on the lights. You can see colors, you can see shapes, you can see borders. You know, if you don't need to wear contacts or glasses or corrective lenses, I can't see any borders of anything when I first open my eyes, but I can see color. So they don't need to build up and turn on slowly. Whereas if you move from a bright room into a dark room, you might know that your eyes adjust. And they take a little bit of time to do that, but when they do, you can start to see more clearly in the dark. So keep that in mind. These rods, they turn on more slowly, whereas a cone, as soon as you're in a bright environment, they're on and they're working. They have different colors inside of them. So there's 3 pigments inside of a cone. We've got blue, we've got red, and we have green. And those help us transmit information about color. If somebody is lacking one of those cones, we call that color blindness, which is way more common in males than in females. There's also an extra special class where mainly females rather than males have an extra cone, and they generally have a red cone, an extra red cone. And those individuals are called super-seerers, and the advantage that they get is they can see many more shades of red than somebody without that extra cone. So super-seerers, extra cone, color blindness, lack of cone. Okay, so the way that we have these photoreceptors respond and send information forward is by utilizing these colored pigments contained in the outer segments. And in both rods and cones, we have these photopigments referred to as opsins. That's our pigment. Again, in cones, we have photopsin, and in rods, we have rhodopsin. And that opsin, it has a shape. It has a shape. It has a conformation. And you might know that things in the body are shape dependent. When these photopigments, when these opsins undergo a conformational change, when they undergo a shape change, they carry forth a signal response. This is your safe place. This is your safe slide. If you're having a hard time in the next few slides when I break it down a little bit more, come back to here. Because what we're talking about is, in this case, we'll be talking about a rod in my example, but we're talking about a resting opsin. It's resting in its bent form. When light hits it, our specific wavelength of light, it causes a conformational change of that opsin, and it moves from its relaxed bent form to its activated straight form. And that is the shape change that's needed in order to get a signaling cascade started. This is what these outer segments look like. So if you look at this picture here, this is a piece of a rod outer segment. And I mentioned that we have these discs that are stacked on top of one another. And inside of these discs of the outer membrane are our photopigments. So here you see the plasma membrane of a cell. We've got our hydrophobic tails, our hydrophilic heads, and that means that the tails, they hate water, whereas the heads, they love water. So this is the inside of a cell, and this is the outside of a cell. And inserted into that membrane is a protein, and if it goes all the way through a membrane, it's called an integral protein, spanning both sides of the membrane, an integral protein. And inside of this integral protein, we have rhodopsin. So this is made up of a couple of different parts. We have our opsin molecules, and we have our retinol. You might recognize retinol because you use it on things like your face, and that's a derivative of vitamin A, and that's really important for helping you see. So anyways, we've got our protein, and inside of it are full photopigment, our retinol and our opsin, to form this big globular protein called rhodopsin. The utilization of rhodopsin as a pigment to transmit information from the rod on is divided into three distinct steps. The first is going to be the synthesis. So rhodopsin has to be brought together. It has to be created. It has to accumulate in the rods, and it does so in the dark. The longer that you're in the dark, the more rhodopsin accumulates in the rods. However, as soon as rods come into contact with light, the second thing happens, which is called bleaching. So that would include the rhodopsin pigment absorbing light, and this opsin protein, which normally exists in this bent form, is going to change into this straight form. And that conformational change is going to be what drives the retinol and the opsin to separate. So we call that bleaching, and you can actually check out bleaching on your own if you're in a room with a light at the top of the ceiling or a lamp. If you stare at that light on the lamp or the ceiling for, 10, 20 seconds, after you close your eyes, you'll notice in the area where the light was in your field of vision, there is just a kind of flashing white thing that you see. It's like a shadow, but reversed. And what that's showing you not only is where that light was in your field of vision, it's showing you that by transmitting information from the rods who had their rhodopsin bleached, their rhodopsin pigment broken apart. So when light is absorbed by the rods, again, that rhodopsin, it dissociates, those two things pop apart, driven by the shape change of this opsin. And as long as they're apart, we're not going to get any action, any activity associated with rhodopsin. If they're apart, it means that you're in the light and we don't need our rods to work at that point in time. So in order to have rhodopsin accumulate and then be active in the rods again, we need it to regenerate. We need those two things, the retinol and the opsin, to come back together. So this is what that looks like here. So here's our rhodopsin, and we mentioned we've got this resting form. So it rests in this bent form. And when light hits it, it converts, it changes to this transretinol. And this is the bleaching process, essentially. And after this happens, and we have, nervous stimulation going to the brain, conveying visual information, not through rhodopsin, which is only going to respond to dark. We want to go from bleaching our rods, bleaching and removing that rhodopsin from signal transmission. We want it to go back to its other form. We want it to go back to the cis-retinol form so that it can get back together with the opsin and then converge back into rhodopsin. So that's the way that this works. It is synthesized. We make it. So we do this opsin and cis-retinol being brought together to make rhodopsin. That happens in the dark. The longer you're in the dark, the more rhodopsin you have. But when light comes in to the eyeball, it will be absorbed and drive this conformational change of the opsin, which is going to pop retinol and opsin apart. And then they need to come back together so that we get more synthesis of rhodopsin. So rhodopsin is broken down in the light and it comes back together in the dark. It works at the individual level. This is what the signaling cascade looks like, starting with this integral protein. And we have our steps outlined here. I'm just going to talk you through them. So we have, again, at rest, our rhodopsin with our retinol and our opsin together. And when light hits it, the cis-retinol is going to move to its activated form. So we're moving from the bent form to the straight form. And that is the conformational change that's needed to move this G protein off of its normal conformation, its normal resting place right here, which is touching. It kind of looks like it's hugging it in this picture. We have that G protein right on the receptor. But when we have this conformational shape change that drives the dissociation of this G protein. This G protein has a special name. It's called transduction, and it carries forth the second messenger cascade. The G protein pops off, and it goes and activates this other integral protein, the main second messenger system used in the visual pathway. It is a protein that is an enzyme. So remember, proteins make up most things in the body, including enzymes. So this integral protein is an enzyme called phosphodiesterase. And remember that enzymes catalyze reactions. They help start a reaction. And the reaction that phosphodiesterase catalyzes is catalyzing the conversion of cyclic GMP to a substance just called GMP. So rather than having cyclic GMP, which is used as a form of energy and also as a gating thing, like we'll see in a second, in this cell, we now just have plain old GMP. Why is that important? So remember, whatever word comes before gated when we're talking about receptors or channels is what opens those channels. So we can have voltage gated channels that open when that cell reaches a specific charge. We can have ligand gated channels. They open when a ligand binds to it. These are called cyclic GMP gated channels, meaning when there's cyclic GMP inside of a cell, like this outer segment of the rod, as long as there's cyclic GMP, it holds this transporter open. And this transporter protein is a pore channel that allows sodium and calcium to enter the cell, to enter this outer segment of the rod. Sodium and calcium are both positively charged ions. So as long as cyclic GMP is around, it holds open these channels. We have the entry of positive ions into the rod, and then we have depolarization. So this would normally be around. Cyclic GMP would be around if there was no light to start this cascade, right? Because if this remained in its relaxed, bent form, we would have none of these other steps, and we would just have cyclic GMP holding open this pore and then positive ions floating in. When we have this conformational change of the opsin proteins, so the rhodopsin, into its straight activated form, we then drive this G protein cascade through that initial conformational change of the shape of the opsin for the G protein to pop off and activate phosphodiesterase, which works to convert that existing cyclic GMP into just plain old GMP. So here's the deal. GMP is not cyclic GMP, meaning it cannot sit here in the receptor, in this channel, and hold open this gate. It cannot open this pore channel. That means the channel will close. So the sodium and calcium that would have gone in during the dark, when there is light, this channel closes, meaning this cell can't be depolarized. The same thing happens with cones, only again, they're active during light, right? So they turn off when depolarization. Okay, it is not rhodopsin inside of cones. It is photopsin. That's your big pigment. And again, there are 3 different types of cones. There is blue, there is green, and there is red. And if you have all of those, you can see the full color spectrum. If you're missing one, then you cannot see the whole thing. You may have heard of red-green colorblind. That is missing one of those, believe it or not, but not both. So again, cones, they go on when it is light. Rods go on when it is dark. The thing that rods and cones attach to, the way that they converge together to form eventually the optic nerve, which would be way down here, is through a series of connections. Why this happens is because rods and cones themselves cannot drive an action potential. They can only drive sub-threshold potentials, which we call graded potentials. So that means you don't get up to the charge you need for voltage-gated channels to open. So when rods and cones either become positive or negative, positive would be depolarized, negative would be hyperpolarized, they send that message on to the next cells in line. So we have rods and cones connecting to the next cells in line called bipolar cells. Bipolar cells have two ends to it and one cell body. There are also some intermediate cells there, the horizontal cells, which work to connect various photoreceptors together to make it to the bipolar cells. So we go rods and cones, horizontal cells, then bipolar cells, and then finally the bipolar cells either are going to use the next level amacrine cells, which run perpendicular to this pathway, to connect to the ganglion cells, or they can connect all by themselves to the ganglion cells. So because they cannot have their own action potential, they can't have that large action on their own, they signal to the bipolar cells and then the ganglion cells. And ganglion cells converge together to form a nervous tract we call the optic nerve. Also, as I mentioned, we have these two intermediate pathways. Horizontal cells come first, and then amacrine cells, which just serve as connecting agents between the photoreceptors or the bipolar cells and the ganglion cells. Just to draw your attention to that is the word glutamate. Glutamate is the main Okay, so now let's put this signal transduction cascade together into what rods do in the dark. And then because rods and cones themselves cannot have their own action potentials, they can only have graded potentials, or what we call generator potentials. That means they can get positive, they can depolarize, but they can't get to the charge needed to open voltage-gated channels to propagate an action potential. Instead, they have sub-threshold, so under the charge needed to open voltage-gated channels, they have those potentials, okay? So in order for action potentials to go forward, rods need to send information, and so do cones, to the next cells in line, which would be the bipolar cells and the ganglion cells. And in this slide, we are not talking about horizontal or amacrine cells. Those are just connection cells. Here, I want to emphasize that rods go to bipolar and then to ganglion cells. Also, for simplification, this picture is a 1-to-1 rod to bipolar cell to ganglion cell ratio, where we usually see many rods to one bipolar cell, in humans at least, because we don't see really well in the dark. But simplification, okay? So what we're looking at is going to affect this final cell, the ganglion cell. And ganglion cells converge together, making their axons turn into a bundle of nerves that together are called the optic nerve. So the optic nerve is the axons of ganglion cells. So they're collecting information from these photoreceptors. So this is in the very back of the eyeball, in the fovea region, where we have this high concentration of photoreceptors. Okay, so let's start with the first thing that we talked about in the signaling cascade, which is what's going on in the rods themselves. So rods themselves, in the dark, their cell type is on. Rods are cells that are active in the dark. And when there is dark, we don't have any conformational change, meaning we don't have the G-coupled protein activated, and we don't have that cyclic GMP turned into GMP. So instead, we just have cyclic GMP, and when that's around, it holds open this channel. It's a cyclic GMP gated channel that allows sodium and calcium to enter into the rod, so the rod itself becomes more positive relative to its resting state. It's about -40 in the dark. Not high enough for an action potential, high enough to do something, high enough to signal the release of neurotransmitter, at least. Okay. So as we have these, of course, there's a dog barking right now next door. Anyways, we have sodium and calcium entering the cell. They're positive ions. The rod itself is depolarized. One thing I want you to see here is at really the synaptic cleft area of every neuron is a voltage-gated channel that lets calcium enter at the level of the cleft. And what calcium does is drive vesicular docking and fusion. Remember, neurotransmitter is contained within vesicles, and those vesicles are made out of cell membrane in the same, phospholipid structure as the plasma membrane. When calcium comes in, it drives these little docking proteins to be expressed on the vesicle and on the membrane. Otherwise, the vesicle will just be floating around. It's kind of like a boat. A boat, there's a sailboat. In order to dock your boat, you need to have a rope or something that connects it to the dock. Otherwise, it just floats away. So vesicles, when calcium comes in, it drives their docking. So these little proteins pop out. And fusion is referring to a vesicle physically fusing with the membrane so that through exocytosis, it can dump its contents outside of the cell. And fusing refers to the fact that this is phospholipid. So as this vesicle attaches to the membrane, it can just be incorporated into the cell membrane itself. Hooray. So that's what we see. Cyclic GMPs around, opening up this gate, sodium and calcium coming in. The rod itself is depolarized. That allows the opening of these calcium channels that are voltage gated. They open when they're about negative 40 millivolts of the charge of the cell membrane. So calcium comes in, which allows neurotransmitter to be released. And that neurotransmitter binds to receptors on the bipolar cell, which in this pathway, this neurotransmitter released is binding to receptors. So even though glutamate is the main neurotransmitter utilized in these pathways, the receptor types on the bipolar cell respond to glutamate in a way that inhibit this bipolar cell. They make it more negative. They hyperpolarize that. And the term for that, IPSP, is inhibitory postsynaptic potential. So very descriptive term. It's inhibiting, it's causing an inhibitory or a very negative postsynaptic, meaning after the synapse, and potential means charge. So when this glutamate is released, it binds to receptors that cause an inhibitory signal. And if the bipolar cell is inhibited, then no calcium is going to enter because we're not at that charge. If no calcium enters, then we're not going to get any vesicular docking and fusion. Without glutamate released here to bind to its own receptors on the ganglion cell that would normally excite the ganglion cell, without the neurotransmitter released and binding to its receptors, we now have the ganglion cell not responding to anything. There's no stimuli to it, so it just kind of hangs out and does nothing. There is no excitatory postsynaptic potential. The ganglion cell is not activated, so there's no action potentials generated in the dark. Which makes sense because when you have light hitting things like cones, that's conveying information about what you see. Like, I see a plant, I see a box, I see a yoga bolster. I'm mentioning all the things that I see right now. And that requires action potential propagation along the ganglion cells, which again form the optic nerve. If I was sleeping and I'm looking at the inside of my eyelids, I don't need visual information going from my rod saying you're looking at dark, you're looking at nothing, you're looking at your eyelids, you can't see anything. Because why? That is an unnecessary transmission of information, and we do not see action potentials then being propagated by ganglion cells in the dark. Don't forget that even though this picture is simplified and we've got rod, bipolar cell, ganglion cell, we also have cones connected. They would be over here, right? So we would have those doing stuff too. So cones also would be connected in this pathway, and they are definitely not sending action potentials on in the dark. However, in the light, we have that change. So here we have a physical rod again. The rod is going to be activated by light so that we get this conformational change of this opsin from its bent form to its straight form. And they're going to break apart and stay that way while they're in the light. So we have that conformational change. We already learned that when that conformational change happens and the G protein cascade occurs, we decrease the cyclic GMP in the cell that would hold open these channels around here to let in sodium and calcium. Without positive ions coming in, we're not going to depolarize this rod. Instead of where it was sitting before at -40, we don't have positive ions come in, so it's going to become even more negative. It will become excessively negative or hyperpolarized. If a cell is hyperpolarized, we're never going to get to the charge we need to open up those voltage-gated calcium channels, which means that no neurotransmitter can be released from the rod itself. Without the rod itself releasing, in this case, the inhibitory neurotransmitter, which we saw in the picture before, it was turning off the bipolar cell. Now we're turning it on. So there is no what we call inhibitory postsynaptic potential. We have an excitatory postsynaptic potential. So we can activate this bipolar cell. It becomes depolarized. These calcium channels are at the charge they need to open. Calcium comes in, drives vesicle docking and fusion, and that then is going to bind to its receptors on the ganglion cell, which has action potentials. That cell can have action potentials. The other ones can just have graded subthreshold potentials. And then those action potentials get together with all the other action potentials from ganglion cells, and they form together in a bundle to form the optic nerves. So this is telling us the rod itself is off in the light, but when it's off, it's not spraying neurotransmitter onto the bipolar cell to turn it off, which would then turn off the ganglion cell. The rod is hyperpolarized, which means the bipolar cell and the ganglion cell can go forth and have action potentials that are related to the cones and the bright vision in the area. Sticking to it, it's the way that it is. All right, so I mentioned this earlier, but it's written out for you here. Light adaptation, it happens really quickly. You don't have to wait for your eyes to adjust. When you turn the lights on from when you were in the dark, they're going to turn on. All the cones turn on at once. You also stimulate the rods at one time and break down all the rhodopsin in them. And it hurts. That's called a glare. And your pupils will change their size. They will constrict to limit the amount of light coming in to be focused on the fovea and keep bothering those rods and cones. So after that glare goes away and you kind of like get on with your life a little bit, in about 5 to 10 minutes, we don't have that really strong sensation. The cones start to level out their degree of graded potential firing. and the rods are slowly turning off and staying off and doing their signaling cascade. So after about 5 to 10 minutes in the light, you see a little bit better because the rods turn off. The retinol sensitivity goes down as we stay in the light, and the cones and the bipolar cells and ganglion cells, they readily adapt. They don't need time to adjust when we move into the light. We just have that initial discomfort brought on by the glare. And then dark adaptation, you have probably experienced this on your own, where when you first turn the lights off, it is impossible to see. You're completely blinded. And that's because when you were in the light, your rhodopsin was bleached. Those parts were, not connected anymore, the retinol and the opsin. So now we have to wait for them to regenerate. They've got to go back to their conformation. And once they do that, they continue to accumulate. So after about, 20 minutes, we see that our vision improves. We've got transducin, that G protein goes back where it belongs, rhodopsin goes back together, and things turn on. And also as an accommodation, the pupils dilate to let in as much light as possible through the eye, focused onto the fovea in the back of the eye. And that, my friends, is phototransduction. It is an interesting pathway where glutamate works opposite and hyperpolarization works oppositely. But if you understand that we don't need action potentials when you're sleeping and looking at the back of your eyelids, but we do need them when your eyes are open and you're looking at stuff, then that should help it make sense a little bit. I don't know. You let me know. All right. Have a great one. Stay smart. Bye.